Hi, my name's Harry and I'm a bioinformatics apprentice working at Lonza Biologics in Cambridge, UK. And today I'll be talking about how we at Lonza are unlocking the secrets of cell productivity using a reverse causal inferencing approach. So just to set the scene a bit, there's a growing need in biopharma for more efficient cell lines producing therapeutic macromolecules. And in order to engineer these more efficient cell lines, it calls for a deeper understanding into the systems that drive optimal cellular performance. So in order to facilitate cell engineering and biomarker discovery, it's key to decode these core systems and interrogate the data. So to do this, we introduced RCI, or reverse causal inferencing, which is a tool that bridges this knowledge gap and provides fresh insights into these fundamental working systems. So then just to take a look at how RCI works. So the input consists of two elements. The first is we take a differential gene expression data set. So on the gene level, it has a fold change and a p-value, and we extract these significant genes based on the p-value and transform them into a quantized state change format. So we assign a directionality based on the fold change, and then we take that significant gene list and also the background genes forward into the next step, which is to combine it with a knowledge base, and the knowledge base consists of experiments in the same format, so significant gene lists in a quantized state change format, stored as a graph, and we take those two elements and perform hypergeometric enrichment analysis to see whether the input data set, the input candidate genes, is significantly similar to any of the experiments in the knowledge base graph. And once we have some results from that enrichment analysis, we can then interrogate it in a number of ways. The most simple way is to just look at the significant experiments and look at their hypotheses, so we can see the treatment and the type of cells and the days in which the contrast and the differential gene expression data was formed. And the second way is that we can cluster together experiments in the knowledge base using TS&E and then perform gene set enrichment and gene ontology analysis on an entire cluster of experiments to see whether there's enrichment for functional gene circuits and physiological systems. And then once we've overlaid that on the overlaid our input DGE or input candidate list onto the TS&E, we can then identify enrichment within clusters and extrapolate into enrichment within physiological systems. So just to quickly go through a case study of how we put this to the test. So the two input elements I spoke about, the knowledge base and the input data set. So we constructed a knowledge base consisting of almost 8,000 public data sets from the Expression Atlas database. And we then took a differential gene expression experiment that we'd compiled from two transcriptomics data sets from a high and low producing cell line. And generated the DGE and then ran the analysis against the knowledge base. And the results that we generated were that we had 173 significant overlaps out of the 8,000 public data sets. And by looking at the clustering of the TS&E and the functional enrichment of the clusters, we discovered that the data set had significant enrichment for secretory functions, immunity, and also proteasome functions. to look a bit more in depth at the results, if you look at the graph in the bottom left, we can see that the TS&E plot and each of the points on the graph is a single experiment within Expression Atlas and within our knowledge base. And the points in bold are the ones that were significantly similar to our input candidate gene list. So you can already see there that clusters 16, 3, and 4 are fairly enriched. There's lots of experiments within those clusters that are similar to our input data set. And then to further look at this, we can look at the graph next to it on the right-hand side, and we can see using another hypergeometric enrichment, we can see that clusters 3, 4, and 16 are indeed enriched from our input candidate gene list. So to finish off, RCI is a way that we can use a list of candidate genes and knowledge base to provide insightful revelations into the active physiological mechanisms that we can in turn use to drive cell engineering and biomarker discovery.